Okay, great. Well, thank you for the introduction. And thank you everyone for taking the time to watch my talk today. So today I'll be telling you a little bit about one of the chapters from my doctoral thesis work, which is also about phage antibiotic synergy. So hopefully all of you got a little bit of a refresher on the topic from Rabia's talk just before the break. And so I'm going to be a little unconventional today, and I'm actually going to start off with the results slide. But I think it's all good because it's kind of a result that all or most of us have probably seen before. Because it doesn't really matter if you're working with Pseudomonas or Burkholderia or Stenotrophomonas or whatever, because it's always kind of similar. And honestly, it's a tale as old as phage therapy. The phage meets the bacterium, the phage kills the bacterium, it selects for subpopulations that are resistant, and those resistant outgrowths are what you see here in green. So these can be receptor deficient mutants, they can be lysogens, etc. And so because this story is actually so common for those of us who work in this field, it's kind of worth asking what actually convinces us that phage therapy will work. And so for me, the answer to that is answered by a single picture. And you might be wondering, why are we looking at a picture of space in a conversation about phage biology? So this right here, what you're seeing is called the Andromeda Galaxy. So it's about 2.5 million light years from Earth. Now, visualize that if we took every single phage particle on Earth and we lined them up end to end, they would actually stretch to that galaxy and back about 21 times. So let's try to kind of wrap our heads around this. There's somewhere around 10 to the 31st phage particles in Earth's biosphere, and it's estimated that they destroy about half of all bacteria every 48 hours. So why am I giving you this very interesting fact? Well, so what this means to me is that phages are extremely successful from an evolutionary perspective, and that's because they're very efficient at what they do. And so initially, this observation can seem to be at odds with our previous observation of, inevitable resistance appearing in our test tubes. But one way that we can reconcile these two ideas is by recognizing that phage-host interactions don't occur in a vacuum. Haha. So bacteria are constantly being exposed to multiple stressors when they're in their natural environment, whether that's environmental stressors like pH or temperature, phages, antibiotics, or metabolites from other microorganisms, predators such as amoeba, and even the immune systems of eukaryotic hosts. And so as the bacteria adapt to resist the strongest of these selection pressures, so for example, in this case, maybe the strongest selection pressure is that green phage that you see, and as the bacteria adapt to resist that specific selection pressure, they can become at least partially more sensitive to others. And so, how can we take advantage of this for phage therapy? How can we take advantage of these evolutionary trade offs? So, broadly speaking, there's two approaches, and we're all very familiar with them because we've actually been discussing them a lot over the last few days. And so, the first of these is sort of, this sort of kitchen sink approach, right? You basically take all the different phages that you have for clinical applications. Maybe you put in some antibiotics or other compounds, you throw them all together and you toss it at the bacteria. And you basically hope that because the bacteria can't adapt to all of these different stressors at once, the bacterial population will be greatly reduced or ideally eliminated. And then the second approach is these sort of guided selection approaches, right? So you can use agents that sort of act in opposite directions on the same trait. So hopefully you guys can see this diagram. It's a little small from where I see it. But basically, the one example of this is using a phage that binds to an efflux pump together with an antibiotic that can only be efflux as a form of resistance. Or another example could be a phage that binds to LPS and a drug that is repelled by LPS. So the bacterial population has a hard time adapting to both of these things at once, and so you can reduce numbers substantially. But so you'll notice that these examples that I gave for guided selection, both of these are examples that deal with resistant outgrowths that are caused by mutational resistance. So what about when the outgrowth is caused by lysogens? So under normal circumstances, as we are all aware, lysogenic phages can form lysogens, obviously, and then they replicate through vertical gene transfer until some kind of environmental cue kind of triggers back induction back into the lytic cycle. So, and we just heard about this from Rabia. So what if we could therapeutically prompt that induction step perhaps with an inducing antibiotic such as ciprofloxacin. So as we just heard, this same idea has actually been explored in depth by the lab of Dr. Alex Hines, who I'm sure is feeling very at home watching this talk. And so his group found in general, to reiterate, that this approach typically tends to produce synergistic killing in E. coli as well as Pseudomonas, and it works in vivo as well as in vitro. However, the authors did note that this approach did not work for every phage bacterium combination that they tried. And so one thing that has kind of remained elusive is whether it's possible to predict using some kind of measurable parameter which temperate phages could synergize with inducing drugs. And so I think at this point, a lot of you may be sitting here wondering, okay, this is great, but why would anyone actually bother researching this? Just go ahead and use a lytic phage. Well, my friends, welcome to the wonderful world of Burkholderia. So this is yet another gram-negative pathogen. And so for those of you who don't know, Burkholderia species cause difficult to treat, often fatal lung and bloodstream infections in people with cystic fibrosis. They're extremely resistant to antibiotics. And to make matters even worse, about 90% of all the phages, and that's not very many phages, by the way, that have been isolated against these species have been found to be lysogenic. So against a pathogen like this temperate PAS would be a very valuable asset. And so we set out to determine whether temperate PAS could actually be developed against Burkholderia. And so we initially started testing this in vitro. So we did this by conducting growth assays with bacteria on their own, incubated with a lysogenic phage, a sub-inhibitory dose of ciprofloxacin, or both of those agents at the same time, which is that blue line that you see at the very bottom there. And so these growth curves were then analyzed using something called the growth reduction coefficient, or GRC. And this is just a normalized metric that uses the definite integral of the bacterial density curve over time. In other words, the area under the curve. And so to demonstrate that synergy could be occurring here mathematically, we ensured that the growth reduction coefficient of the combination, which is shown in that blue bar there, the dark blue bar, is significantly greater than either of the agents on their own, shown in light blue and yellow, but also that it's significantly greater than what we see in that gray bar, which is the GRC value that we would theoretically expect to see if the effects of the individual agents were simply stacked on top of each other. In other words, this would be a mathematical effect. Sorry, an additive effect. So the additive effect is what you see in the right-hand side of that inequality. And so this is the theoretical value that we would expect to see. And then the left-hand side is what we actually see. So if this inequality is satisfied to a significantly meaningful degree, then we can say that mathematical synergy is occurring. And so we actually repeated this approach on many different combinations of Burkholderia species, many different lysogenic phages, and we found that in many cases, temperate phages do in fact synergize with sub-inhibitory doses of ciprofloxacin. So this is possible for Burkholderia, but not all of them. So there were definitely cases where no synergistic effects were being observed. And so we kind of just went back to our previous question, which is there actually a parameter that we can use to predict which phages will synergize and which phages won't? Now, in a previous study, we introduced this concept called the stable lysogenization frequency, which is just a straightforward metric that essentially subcategorizes phages, subcategorizes lysogenic phages. And so this is simply the proportion of survivors of a phage X that are lysogens of that phage after some time t, under a set of environmental parameters that include medium, MOI, temperature, and the host strain. And so you could probably add other parameters as well, but these are the ones that we tested in the study. And so what we observed is that when you hold all of these different variables constant, the lysogenic phages in our data set sort of broadly partitioned into these highly lysogenic phages that had very weak antibacterial effects and very lowly lysogenic phages that had strong activity. So that activity was actually equivalent to a lot of obligately lytic phages that we added as comparisons. And so in fact, we saw a very strong and significant inverse correlation between lysogenization frequency and the growth reduction coefficient. So in the present study, we replicated the exact same result with a smaller data set, but then we showed that this correlation is completely abolished in the presence of sub-inhibitory doses of ciprofloxacin. And so this suggests to us that ciprofloxacin can actually correct the defect in antimicrobial activity that's caused by lysogeny. And so simultaneously, we observed that the magnitude of the synergistic effect is directly correlated with the lysogenization frequency of the phage that's being used. So in other words, the more lysogenic the phage is, the weaker the effect is on its own. And so the more the combination can improve the effect, and so the stronger the synergistic effect becomes. And so this tells us that this form of synergy is specifically tailored to highly lysogenic phages that are ineffective on their own. And so at this point, we have evidence that temperate Burkholderia phages can synergize with subinhibitory ciprofloxacin, and that this actually occurs in a lysogenization dependent manner. So the more lysogenic, the more it works. But we wanted to explore the potential mechanism. And so in particular, we wanted to explore whether induction of lysogens could be responsible for the synergistic effects that we're seeing here, just as they were when in TPAS that was targeting E. coli and Pseudomonas. And so here we focused on phage KS5, which was targeting Burkholderia type strain C6433. So this phage had one of the strongest synergistic effects recorded with ciprofloxacin. And so what we noticed is that lysogens made up about 75% of the KS5 survivor population when the phage was administered on its own, but this decreased by more than tenfold in the presence of subinhibitory ciprofloxacin. So this tells us that the drug is somehow depleting stable lysogens of this phage. How is this happening? So finally, we observed that KS5 lysogens were more sensitive to the effects of ciprofloxacin, and this was shown both in microdilution assays, which is shown in the main figure here, and in Kirby-Bauer inhibition assays on plates, which is shown in the inset. And so together, these data sort of suggest that KS5 is normally making the lysogens, but the prophages are hypersensitive to ciprofloxacin and are induced, which basically depletes those lysogens. And together, this results in a synergistic killing effect. So we can't say with 100% certainty that this mechanism occurs in all of the tested cases with Burkholderia temperate phages. But in this specific case, we feel that induction is the most likely explanation behind the effect. So to summarize, ciprofloxacin is able to overcome antimicrobial limitations caused by lysogen formation. And so this means that lysogenic phages could theoretically be used as adjuvants to standard antimicrobials. And finally, this gives proof of concept that TPAS is possible with at least some lysogenic Burkholderia phages, which is great news for patients suffering from Burkholderia infections. So all of the data that I've shown here has now been published in scientific reports. So please feel free to check it out if you want to read more on the topic. Personally, I find that I'm a much better writer than a speaker. So if I didn't convince you here today, maybe the paper will. But with that, I would like to thank all the funding agencies that made my PhD work possible. And of course, the organizers of Phage Canada for putting this all together. And finally, I'd like to thank all the members of the Dennis and Ravio labs that I worked with over the years of my PhD. And with that, I'd be happy to answer a few questions.